Okay, we're going to get going on our next topic. So the next thing that we're going to start talking about is the cell membrane and transport within the cell membrane. Okay, so hopefully you remember that when we just talked about organelles, one of the organelles that we did discuss was the cell membrane, and its job is to control what goes in and out of the cell. Okay, and so in order for us to be able to look at that and understand how things move into and out of a cell to help the cell stay healthy and stay alive, there's a few terms that you guys have to understand before we start doing that. So I'm just going to start this off by us talking about what these words mean so that when I use them while I'm teaching you about the cell membrane, it makes more sense to you. Okay, so the first thing is the idea that when you're talking about molecules moving into and out of a cell, okay, molecules are going to move based on something called concentration. Okay, so what we're going to do is we are going to talk about a couple of different scenarios of what concentration is and then apply it to the cell membrane. Okay, so the word concentration itself, scientifically speaking, is basically when you're focusing on the amount of solute in a location, okay? And so when I use the word solute, hopefully you remember from middle school that there are different terms, okay? Solute and solvent, okay? And so when we're in school, I always tell my students to think of it like if you're gonna make, I like to drink the tea that you make from the little tea packets. And so the solute is going to actually be like the little tea powder. Okay, and then the solvent would be the water that you mix with the tea packets. Okay, so hopefully you know these two terms. Solute, again, is like whatever molecule is there, and solvent is what it's dissolved in. Typically, it would be talking about water. Okay, so when we talk about this word concentration, we're going to reference high concentration and low concentration. Okay, so typically when you're talking about high concentration, okay, when we do this in class, I always have my students draw pictures because it's usually so much easier to do that. When you're referring to something that has high concentration, that means there is a lot of solute. Okay, and so I have my students draw it, and you can always know when we're talking about cell membranes and transports in class because you're always going to hear this tapping sound. Right, so when there's a lot of solute, and each one of these little dots I'm making is a molecule, right? You can think of it as like a little tea packet. It could be a piece of salt. It could be a piece of glucose, okay? Anything other than what it's dissolved in, okay? And so when we're talking about this, there is a prefix that you are going to use, and the prefix that we use to describe when there is a lot of something is the word hyper. Right, and I always tell kids or students that if you're thinking of the word hyper, hopefully you think of somebody that has a lot of energy, right? They're hyperactive. They have high amounts of activity. Okay, so we'll use the word hyper in order to describe a lot of things, and then because we're talking about the solute, we'll usually use the word tonic. Okay, so whenever you have a place that has a lot of stuff, we refer to it as hypertonic. Okay, you can alternately have something that is low concentration. Okay, so in this case, you would be talking about less solute. Okay, so in those cases, you would be talking about less. The prefix we are going to use will be hypo. And again, because we're talking about the actual solute, we're going to use the word tonic. Right? So in locations where there's less stuff, we would refer to it as hypotonic. Okay, so the thing you have to keep in mind, though, is always a comparison. Okay, so if I drew something, let's say if I had these situations, right? If I have, let's say I have a solution that has this much, right? But then I have a different solution that's right near it that has this much. Hopefully you could see that this one we would label with an H because it's high concentration compared to this side that has low concentration, right? Because it's a comparison. So I would refer to this side as the hypertonic side, and I would refer to this side as the hypotonic, right? So this one would be the hyper, and this one would be the hypo, right? And instead of doing pictures like this, I could give you like percentages of things, right? Like let's say this side had 17% oxygen, and this side had 6% oxygen, right? This one has more than that one, so this one would be our hyper, this one would be our hypo. Okay, but now because it's again about comparison, let's say you're comparing to that, right? So if you look at this box now compared to this box is no longer low because we're comparing it to this one, right? So this one is no longer our low side, and I guess I could just go ahead and erase it, right? That side is no longer our low side. Now this is our high, this is our low, right? And so I would refer to this as my hypertonic, and this would be my hypotonic. Okay? So these are just terms of things that you guys need to make sure that you know in order for us to be able to talk about what we're going to talk about. Okay, so moving on. Hope I don't run out of pages. Okay, so now back to the cell membrane itself. When you talk about the cell membrane, okay, it controls what goes in and out. Don't know what happened to my end. Right, it controls what goes in and out of a cell. Okay, and by doing that, it keeps the cell alive. And in order to do that, it's made of these little things called phospholipids. Okay, and so what one phospholipid looks like, it's got a little head, and they call them tails, but for some reason I can't get tails in my brain. I always call them legs, but whenever I refer to legs, I'm actually talking about the tails. Okay, so in these, this head part is what we refer to as a phosphate head. Okay, and then the legs are lipid tails. Okay, and so this part up here, the phosphate heads, they love water. Okay, and these lipid tails, just based on the chemical structure that makes them up, they hate water. They're afraid of it. Okay, so what ends up happening is this being one phospholipid, when an actual cell membrane is made, many of these phospholipids come together. Okay, and so I'll just start out by drawing the little heads. Okay, there's the little heads and there's their little legs. Okay, there's a whole bunch of them. They all smush together to make this like membrane. Okay, but we just said this top, the little heads, love water, and if this is in a cell, it's floating around in a whole bunch of water. But these little legs, they don't like water. They're afraid of it. Okay, so what happens is there is a second layer, okay, but it's flipped upside down. Okay, and it should be two little legs on each one. Okay, so this is, now you can imagine, I'm not going to draw the whole thing. Now imagine you have a situation where that is our cell, right? So I just drew a little bit. So imagine this whole line that's going around is made up of all little phospholipids like that, okay? And it is referred to as a phospholipid bilayer, right? Phospholipid because it's made of many phospholipids, bi meaning two, layers meaning layers, right? So it's a two-layered membrane. Okay, so this is the cell membrane, okay? And based on this, there are certain things, let's use pink, right? Let's say something wants to get into the cell, right? So let's say we have this little pink particle, it is floating up to the cell, and it wants to go in, okay? There are certain criteria that it either has to have to be able to pass through, or it has to find a different way in, okay? And then the same thing can be true when we're talking about transport. It's not only things trying to get in. Let's say that there's something here inside of the cell that wants to try to get out, okay? So we're going to talk about the ways that those things can try to move in and try to move out. Okay, so when you are trying to have things move, the natural movement of molecules is going to be what we refer to, and I always shorthand it, from what we call high concentrations, so the side of the cell that has more solute, and the molecules will move from there to the place where there is less solute. Okay, and so we refer to this type of movement, it is called diffusion. Okay, so for example, let's say you have a cell. Okay, so here's my little picture of my fancy cell. Okay, and let's say on the inside of the cell, let's say we're talking about oxygen. Let's say out here, there is 25% oxygen. Okay, inside the cell, let's say there's 7% oxygen. And so oxygen is going to want to go from where it's in high concentration to where it's in low concentration. And that high concentration is out here, right? 25 is higher than 7, so I would label this as my high concentration. The 7% is less than 25, so I would label this as my low concentration. So I know that those oxygen molecules would cross and move across the cell membrane and go into the cell. Okay? Alternatively, you could have another cell or the same cell. Okay, and instead of talking about oxygen, let's say we're talking about carbon dioxide, right? This one over here, this is O2, right? It makes sense. You want oxygen to get into your cells. Let's say over here, we're talking about CO2, right? Let's say that there's 17% CO2 inside the cell, 3% outside the cell, and I'm just making these numbers up, right? So we know just simply based on the numbers, this is our high concentration, this is our low concentration, so the carbon dioxide molecules will leave and go out of the cell. Okay, and both of them are going to move until they are balanced, right? Until the percentages are equal. Okay, and that is going to be a state which we refer to as dynamic equilibrium. Okay, so again, diffusion is going from high concentration to low concentration, and you have to identify where is the high and where is the low, and those particles will move from where they're high to where they're low. Okay. And they will cross the cell membrane to go to the place where they are low because they're trying to balance everything out, Okay, that is a process called diffusion. Okay, so that process of diffusion requires no ATP, right? ATP being, oops, if I could spell, ATP is energy, right? So that's why I referred to it as natural movement. This just naturally happens. Okay, so when you're talking about molecules moving from where they're high concentration to where they're low concentration, okay, there are a couple of different ways, oops, that this could happen. Okay, so we are going to talk about, there are two types of passive transport. Right, again, passive meaning no ATP. Okay, and so this is going to be when things move from high concentration to low concentration. Okay, so the first one that we are going to talk about is one called simple diffusion. Okay, so again, with our cell, okay, here is a cell. Okay, and again, think of that cell membrane that I just drew being made of a whole bunch of those little phospholipids, right? A bilayer of those things. We just simplify it and draw it like this. Okay, so what this cell membrane is, and here actually, let's just do it that way. It might make it easier on you. Okay, I'm just going to draw the cell membrane, just a portion of it. Okay, but you can assume that this is like a whole cell. Okay, and then I get to the point where I kind of draw these fast. So please remember that there's a head and two tails for each phospholipid. But for the sake of getting you through your notes, we'll just draw it quickly like that. Okay, so simple diffusion, okay, this is going to be movement when the molecules squeeze between, I abbreviate between B/T, between the phospholipids. Right? So like there's little tiny spaces here, and so let's say we have a little molecule out here on the outside of the cell, and it literally is just going to squeeze down in here between and come out the other side. That was weird. Okay, so it's simply going to squeeze between those phospholipids. In order to be able to do that, in order to be able to fit between those phospholipids and allowed to be able to pass, they have to be, the molecule that's trying to pass has to be small and I have to emphasize the and, neutral, meaning it cannot have a charge. Okay, so things that can do this would be things like oxygen, carbon dioxide, right? They are both small and neutral, so they can just squeeze between. If something is small but it is not neutral, it cannot pass this way. If something is neutral but not small, it cannot pass this way. It has to have both of these criteria. Okay, so that is simple diffusion. Okay, so again, going from where it's high concentration to where it's low concentration does not take energy. Okay, and it squeezes between the phospholipids. It has to be small and neutral. And the examples are oxygen and carbon dioxide. Okay, so again, we call that form, that one is called simple diffusion. Okay, so the next kind, let's choose a different color. Let's go with orange. The next kind is called facilitated diffusion. Okay, so both of them are types of diffusion. Okay, so because they are both types of diffusion, the same thing holds true, right? This is still going to move from where the molecule is in high concentration to low concentration. Okay, and it also is not going to take ATP, right? No energy is needed for this to happen. Okay, so how this is different is this. Over here in simple diffusion, we said that if you are small and neutral, you can pass this way. But not everything is small and neutral that needs to either go into or out of your cell, right? So there has to be another way for things to get across. And the other way is this one called facilitated diffusion. So here we are with our little cell membrane again. Okay, and so I'm going to draw the little heads. Okay, but let's say there's something that's really big that needs to be able to pass through. Okay, so for like example, glucose, right? The chemical formula for glucose is C6H12O6. Okay, that's a pretty big molecule compared to CO2 and O2, right? There's a lot of atoms that make this thing up. So the guy is pretty big. So in order for it to pass, there are these proteins that are embedded inside the cell membrane. Okay, and so here's my cell membrane. Right? This C6H12O6, this glucose, is way too big to be able to squeeze between these phospholipids. Right? It's like I always tell my students, it's like me trying to get an elephant in through my classroom door. It's just not going to work. Okay? So you have these proteins that act as a larger pass-through to get these bigger molecules into or out of the cell, right? So what they do is, let's say that you have glucose floating around out here and it's a big guy, right? He's not going to be able to squeeze between. So he can actually pass through in between this protein, right? So you need to make sure you know this is a protein. Okay. Sorry, don't know what happened to my pen. Okay, so this is a protein that allows it to happen. Okay, so you can call this facilitated diffusion. This is a carrier protein, right? And there's different kinds of these depending on how they actually work. Some of them like move and let things through like a sliding door or a revolving door. Some of them are just constantly open, called channel proteins. But for the most part, I just want you to know that facilitated diffusion is when there is a protein that is embedded that allows things to pass. Okay, and this is the way I always tell students to remember, because this one rhymes to me. The things that can pass this way are large and charged. Right, so anything that is large or anything that has a charge to it. So we already gave the example of glucose being something that's large. So something that has a charge to it is like something that's an ion, right? So like sodium with a plus. That little plus up there tells you that it has a charge to it, okay? Potassium, chloride, chlorine, right? So all of these things, or anything that is polar, right? Anything that has a partial positive and partial negative charge to it are going to pass this way, okay? All right, so just to recap them, so you got them straight in your head. Simple diffusion is moving from high to low. No ATP is required. Okay? And it is when things are small and neutral, and they can squeeze between these phospholipids and either go into the cell, if that's the direction that the high to low tells it that it needs to go, or it can go out if it's high inside and low outside. It can move out that way. Okay, but it has to be both small and neutral. Okay, facilitated diffusion is also something moving from high concentration to low concentration. No ATP or energy is required, but because the things are either big or charged, they're not allowed to pass between the cell membrane like they did in simple diffusion, right? So there's these proteins that are embedded in it that act as like a tunnel through a mountain that lets the things pass. Okay, so I'm going to read off to you a sample problem And then I'll draw the picture that I would have you guys draw if you were in class to help you figure out how you would want to work this kind of problem. Okay, so one of the questions that I have, a sample problem that we would have done in class, it says a patient in the hospital has 3% glucose in their cells. So I always tell my students, please do not try to do these in your head, right? Draw a picture of a cell. And we said that inside there is 3% glucose. Okay, that is 3% inside, and they are given a solution that has 10% glucose in it, right? So that is going to be something that is given to you that will get into your blood, that will be outside of the cell. So you're going to have 10% glucose out here. Okay, so the questions that I have on here, it says, using dots as glucose, draw a picture that represents this scenario, right? So here is the scenario if I do it in percentages. If I was going to draw dots, which we will do a lot of when we get back, right? I have less dots inside, right, showing that it's low. And then outside, there's going to be a lot more. All right, so this is what I mean when I say draw a picture. All right, lots and lots and lots outside. Okay, and then I always tell my students, and it's just a habit that I try to get them into, label where the less concentration or the low concentration is with an L, put an H on the side that's high. Okay, so then the question would be, what direction do you predict the glucose will move? Okay, so remember we said it is going to move from where it is in high concentration to where it's in low concentration. Okay, so in this case, it would move into the cell. What adjective would you describe the glucose solution, right? And out here is the solution compared to the cell, okay? And so this goes back to what we talked about in the beginning about the naming of it, okay? So remember out here where it's high, that is what we would refer to as, let me change the color so you can see it, right? High concentrations, this is going to be hypertonic. And then inside where there's less, that is what we refer to as hypotonic. What the heck? Okay, so this is my hypertonic area. This is my hypotonic area. And then the last one says glucose is a large molecule. How will it pass through the cell membrane, right? And so this is basically asking, is it going to pass simply or is it going to pass facilitated? Right? And so we said glucose is large, right? So that goes along with the large and charged. And so this is going to pass through facilitated diffusion. Okay. All right, so I hope that helps. Make sure you take pictures of these or actually upload these, and then you can move on to your next task. Good luck.